Today we're going to be talking about Chapter 4, Sections 4 through 6. This will be an overview of cellular respiration as well as some of the details of the process and also the process of fermentation. So before we begin, make sure that you have something to take your notes on. The Power Notes sheet for this section looks like this. You have some diagrams as usual to label. And then also importantly on the back is a summary chart that I will encourage you to fill in after we discuss respiration. And then at the very end, you'll see information about fermentation. So if you don't have this printed out yet, I encourage you to pause the video, go get your Power Notes sheet, and then return. So our key concept for cellular respiration is that we are using this chemical process to convert sugar in the form of glucose to ATP. And when we're talking about cellular respiration, the default is to assume that we're talking about the kind that uses oxygen, which is called aerobic respiration. And you can see the equation for this given at the bottom of the screen here. So we have it both in pictures so you can see what the molecules look like with their chemical formulas, and then just the chemical formula here at the bottom. Now when you look at this equation, what you will notice is the reactants are glucose and oxygen, which were our products from photosynthesis, and the products of cellular respiration are carbon dioxide, water, and energy. And remember, those were the reactants of photosynthesis. So essentially, photosynthesis and cellular respiration are going to be a constant cycling of matter, where matter produced by photosynthesis is then used as a reactant for cellular respiration. Now, the energy is what is different. The energy in the form of photosynthesis is input as light and stored as chemical energy, and then in cellular respiration, that chemical energy that's stored in glucose is able to be released. And that's why you see energy in the form of ATP is a product of cellular respiration. So essentially, all cellular respiration is the process of making ATP, and we do that by breaking down sugar. There are three main stages for cellular respiration. They are glycolysis, the Krebs cycle. In some sources, you'll see this called citric acid cycle because citric acid is produced as a part of the cycle. However, the materials that I produce, I just call it the Krebs cycle. So that's the term that you can remember. And then the third main stage of cellular respiration is going to be the electron transport chain. We've already learned about electron transport chains previously, and so you're going to see that similar idea here. Now, one thing to know about glycolysis that is really important is that it occurs in every single organism on Earth, even prokaryotes. Bacteria can do glycolysis. Animals do glycolysis. We have glycolysis happening in our cells right now at this very moment. Plants do glycolysis. Fungi, even a single-celled pond organism is going to do glycolysis. So glycolysis is a very key metabolic process that happens in all living things, and that means it doesn't need any particular special structures to take place, and also it can happen in the absence of oxygen. Oxygen being present doesn't prevent it from happening, but it can continue even in environments where there is no oxygen. Now, those last two stages, the Krebs cycle and the electron transport chain, do require oxygen. So we call them aerobic stages. Aerobic just simply means it requires oxygen, and these stages do require complex structures. They do require the membranes of the mitochondrion, which you can see in the image here. So after we get past the glycolysis stage and we get into the second two parts of cellular respiration, we're talking specifically about processes that do require oxygen and do require the specialized structure of the mitochondria. So speaking of a mitochondrion, let's take a look at what that structure looks like. You can see this image here on the diagram. You have on your Power Notes sheet an outline of a mitochondrion, so I recommend labeling the parts on your sheet that you can see here in the image. So some of these important parts would be, first of all, it consists of both an inner and an outer membrane. You can see them both labeled in the image here. The outer membrane is smooth. The inner membrane is actually highly folded. The inner membrane is larger than the outer membrane, so it's all folded up to be able to fit inside of that outer membrane. That's going to increase the surface area of that inner membrane because, you can see this in the picture, on that inner membrane, we have all of these little knobs. These are ATP synthase molecules. So remember that from our discussion of ATP earlier in this unit. ATP synthase is located all throughout that inner membrane, and therefore having a large amount of inner membrane allows mitochondria to produce a large amount of ATP. The inner membrane is folded, as I mentioned. Those folds have a specific name. They're called cristae. And so you can see them labeled in the diagram here, and that would just represent a fold within that inner membrane. We mentioned ATP synthase being found in the inner membrane. And if we look out of that inner membrane, in between the inner and outer membranes, we have the intermembrane space. The intermembrane space is going to be important because that's where hydrogen ions are going to be building up in the mitochondria. Remember that ATP synthase is powered by hydrogen ions. So we have to have a compartment in the organelle where we can build up that hydrogen ion concentration. That compartment is the intermembrane space. The interior part, so inside of that inner membrane, is a fluid. Just like in a chloroplast, we called the inner fluid the stroma. Well, in a mitochondrion, we call it the matrix or the mitochondrial matrix. And again, it's that interior fluid. It's a jelly-like fluid, kind of like the cytoplasm of a cell. And also, interestingly, we'll talk about this in our next unit, mitochondria have their own DNA and ribosomes. So mitochondria can actually make things that they need on their own without relying on the larger cell nucleus to dictate how to make those things. The ribosomes are making proteins inside of the mitochondria. All the proteins needed for cellular respiration are made right there inside of the organelle using its own ribosomes. So let's look at glycolysis, which is that first stage of cellular respiration. This involves the splitting of glucose into pyruvate. Pyruvate is also called pyruvic acid. If you're thinking of the POGL that we did in this unit, pyruvic acid and pyruvate are the same thing. Now, this is going to result in the net production of 2 ATP molecules. So what does that mean? We actually have to use 2 ATP molecules to split the glucose, but the reaction that happens after we split glucose is producing 4. So if we have to put 2 in and we get 4 out, that is a net production of 2 ATP. This is also going to produce 2 molecules of NADH, which is an electron carrier like we learned about in photosynthesis. It's also carrying that H, that hydrogen, so it carries both electrons and hydrogen. In the POGL, it talked about NADH being a hydrogen carrier, so it carries both hydrogen and electrons. And the end result of glycolysis is going to be 2 molecules of pyruvate. So this is what a diagram of the process would look like. I'm not going to have you recreate this diagram by memory at any point, but you do have this diagram on your Power Notes sheet. I do recommend that you label the portions that are blank on your sheet just so that you can reinforce what is being used during the process and what is being produced during the process of glycolysis. So let's talk about these electron carriers a little bit more, because This is a really important idea. We're going to start with NAD+. This is an electron carrier, so it is able to pick up electrons and carry them to ultimately the final stage of cellular respiration, which you remember from earlier is going to be the electron transport chain. Something called the electron transport chain is clearly going to need electrons. So NAD+ is going to pick up electrons and carry them to the electron transport chain. In the process, it turns into NADH. Now, we've discussed this previously in class, this idea of a molecule picking up 2 electrons and a hydrogen ion to become another molecule. In photosynthesis, this was NADP+ that picked up 2 electrons and a hydrogen, but in cellular respiration in animals, we don't have NADP in our cells, we have NAD. It's a similar molecule, and I'm giving you the molecular structure here, not so that you can memorize it, but just to show you this is what we're starting with. You can see we have a positively charged nitrogen to begin with. That's where the plus comes from. So this is able to pick up 2 electrons and also a hydrogen to turn into this molecule over here. So you can see the extra hydrogen is now located on the top of the molecule. So that's where the change has occurred. Now, there's a second electron carrier that we're going to talk about in cellular respiration. This one is called FAD. It can also pick up 2 electrons, but because FAD doesn't start with a charge, when it picks up 2 negatively charged electrons, it has to pick up 2 positively charged hydrogen ions to balance out its charge. So it turns into FADH2. Again, you don't need to memorize these structures. I'm not going to show you a picture of either one of these molecules and have you identify it from memory. I just wanted to highlight here what these molecules are doing is that they're picking up electrons and hydrogen, and they are carrying those things into the later stages of cellular respiration. And that later stage of cellular respiration, where most of those electrons are used, again, is going to be the electron transport chain. The second stage of cellular respiration is going to be the Krebs cycle, but before we get into the actual Krebs cycle itself, that pyruvate from glycolysis needs to be processed a little bit further before it can enter into this Krebs cycle. So it's going to actually release some carbon dioxide as those carbons are being broken off. It produces some more NADH in the process, and that broken down pyruvate at the end is now ready to enter into the Krebs cycle. So you can see down here, and don't worry about coenzyme A or any of that. I'm not going to ask you those names, but this pyruvate that came in is continually broken down. Now it's in a form that can enter into this Krebs cycle. So we will talk about the Krebs cycle on the next slide. Your big takeaway message for the Krebs cycle is simply its purpose is to produce energy carrying molecules. So if we look at it, it is a cycle, and don't freak out when you see this diagram. I'm not going to ask you the details of the diagram. What I really want to highlight here are those energy carrying molecules. And what I mean by that, I mean the NADH that's produced. NAD+ is picking up electrons, which are a source of energy later on in the electron transport chain. We see the production of ATP. So this process actually rebuilds ATP from ADP and inorganic phosphate. And then in a third spot down here in the bottom, we see more NADH and FADH2 being produced. Again, those are both electron carriers. They're going to carry those electrons to that final stage of cellular respiration. So now let's look at the specific steps of the Krebs cycle. Here's that same diagram that we just looked at. So to summarize what has happened here, that intermediate molecule, we said the pyruvate has to be broken down a little more before it can enter the Krebs cycle. That's what I mean by the intermediate molecule. So that enters the Krebs cycle and it goes through a series of reactions. That's all you need to know about the reactions. Don't worry about how many carbons or what's formed or how many molecules. As those molecules get broken down, we do have CO2 continue to be released as a waste product. We're breaking off those carbons. We're trying to strip the electrons so that we can make NADH and FADH2 and use those electrons later on in the electron transport chain. So those two electron carriers are doing their job of picking up the electrons. As we're breaking off carbons, that is going to make electrons available for NAD and FAD to be picking up and carrying with them. And then finally, we also do have the production of 2 molecules of ATP in the Krebs cycle. So that brings us to our electron transport chain. This is the third and final part of aerobic cellular respiration. And the point of the electron transport chain is to use those electron carriers. It uses NADH and FADH2 to make ATP. The electrons from those electron carriers are going to enter into the electron transport chain. And as they move through it, the energy from that electron flow is going to pump hydrogen ions across the inner membrane. Remember, we've already learned about electron transport chains as a part of photosynthesis. So the same general principle of using electron flow to pump hydrogen ions across the membrane is still true. Then those hydrogen ions, once we have them all built up in one area of the mitochondria, they have built up into a high concentration. And what do we know about hydrogen ions that have built up into a high concentration? If there is an ATP synthase, they will then go through ATP synthase, and the energy of hydrogen moving through the molecule turns ADP back into ATP. So again, look at these electron carriers. We have our NADH, we have our FADH2. What are they doing? They are giving their electrons to this electron transport chain. The electron transport chain is pumping hydrogen ions into the upper part of this diagram here above the membrane. They're building up into a high concentration so they can go through ATP synthase and turn ADP back into ATP. Now, what are these hydrogens going to do once they come across the membrane? And what are these electrons going to do when they get to the end of the electron transport chain? Something has to pull them out of the chain so that more electrons can follow behind them. And that something is going to be oxygen. Oxygen is responsible for picking up those electrons and the hydrogens. And when it does that, it produces water. So let's look at that in a little more detail. Here is the same diagram. It shows the proteins. These circles are proteins that are embedded in that membrane that are part of the electron transport chain. NADH is dropping off its electrons and turning back into NAD+. FADH2 is dropping off its electrons and turning back into FAD. And then finally here at the end, we have those electrons. This is the path of electrons moving through the electron transport chain. Those electrons are being picked up at the end by oxygen. Remember when we talked about water, we said oxygen is very electronegative, meaning it likes to pick up electrons. It is a great thing to have oxygen at the end of an electron transport chain because it is so attracted to those electrons. So when it picks up those electrons, it picks up some hydrogen ions as well and turns into water. So water is one of the waste products of cellular respiration. This idea here of oxygen being at the end of an electron transport chain, and what it's doing is it's taking those electrons, there's a term for that, and we would call that a terminal electron acceptor. Why do we do that? Well, it's accepting electrons, and it's at the end. Terminal means at the end of that electron transport chain, so oxygen is our terminal electron acceptor. It pulls electrons, it pulls hydrogen ions, and combines all of that together and forms water. At this point, I'm going to remind you of that summary chart in your Power Notes sheet and encourage you to go ahead and fill this out and summarize those 3 stages that we just discussed. Now, once you have that summary chart done, let's move on and talk about fermentation, the last part of this presentation today. The main idea for fermentation is that this is simply going to allow glycolysis to continue. Fermentation itself is not going to make any ATP, but if there's not enough oxygen available for aerobic cellular respiration, we need to somehow recycle that NADH back into NAD so that glycolysis can continue. So fermentation will allow that process to happen. As I mentioned, this is anaerobic, meaning it does not use oxygen. And we can even notice this in our cells. If we are exercising and using a lot of energy, breathing a lot of oxygen, if we are breathing heavily, that means our cells want more oxygen so they can do more aerobic respiration. So if they aren't getting the oxygen that they need in order to make the ATP, they start the process of fermentation. So fermentation itself does not produce ATP, as I mentioned. It is simply going to allow glycolysis to continue. So you can see glycolysis with the presence of oxygen is going to be followed by cellular respiration that we've just discussed in the previous part of this video. But if there's not enough oxygen available, then glycolysis leads directly to fermentation. So let's talk about the 2 types of fermentation that can happen and what kind of organisms they happen in. Essentially, we have alcoholic fermentation as one type, and we have lactic acid fermentation as the second type. They're both important for organisms to be able to make ATP in low oxygen or 0 oxygen environments. Alcoholic fermentation is something that is done by yeast and other microorganisms such as bacteria, and it forms alcohol, as it says in the name, and carbon dioxide as waste products. So if you have ever baked bread before, you know that you have to put yeast in there and you have to let it rise. That's because the yeast are fermenting the sugar that you put in the bread dough, and then the rising comes from the carbon dioxide that's being produced as waste. It does actually produce alcohol as the yeast are making your bread dough rise, but it's such a small amount, and when you bake it, you bake that alcohol out, and so that's why our bread is not alcoholic. Alcoholic fermentation is also used in brewing alcoholic beverages such as beer and wine. So this is a very important industrial process using yeast to ferment things in the baking and in the alcohol industry. The second type of fermentation, lactic acid fermentation, is actually happening in our bodies whenever we are running low on oxygen. It's used by our muscles, it's used by a lot of other unicellular organisms, different types of bacteria, and it produces lactic acid as a waste product. So as we were mentioning earlier about doing exercise and breathing heavily, you probably notice when that happens, your muscles start to burn. That's because your muscle cells are trying to make as much ATP as possible. If they're not getting enough oxygen, they're going to resort to lactic acid fermentation, and this causes a buildup of lactic acid in your muscle. Now, as you can imagine, if you have acid building up in your muscles, you're going to feel that, and that's where the burning sensation comes from. Your body is able to break it down, and it's not harmful to your muscles, but if you've ever wondered why your muscles start to burn when you exercise, that would be why. They are doing lactic acid fermentation. So the fermentation section on your Power Note sheet is at the very end. It's just a short summary based on what we just talked about. You don't need to know a lot of details about fermentation, but do make sure you have that written down. And so that is the end of Chapter 4, Sections 4 through 6. Hopefully you found this to be helpful, but if you still have questions, please make sure you ask them in class.